Hi, so what we're going to be doing in this video is navigating the main features of PyShifts, as well as just how to navigate the platform in general. So I've gone ahead and done is I've moved into a temporary location on PyMorg, and I'm now going to fetch a coordinate set from the protein data bank called 2KOC. So if I type that in, I can now see that it pops up, the object pops up on my PyMorg. Now, in order to actually analyze this, we're going to need to type this object into PyShifts as well. So I'm going to be typing 2KOC. And now we can see below there's a mode box with Lamore D, which is what we're going to be using with this specific one. Lamore D is the mode we use for RNA. Lamore CA is the one we use for proteins. And Lamore, or other, my bad, is the one we use when we bring in our own personal chemical shift files. And so we're now going to be running this platform, which may take a second. We can now see that the error, we've moved on to the error analysis section. Now, in order to actually analyze this object, we're going to need to bring in a chemical shift file. And I'm going to be doing that by clicking the Browse button and moving to my PyShifts folder. Or we can see that we have a measured shifts file called 2KOC. And so this is the file we're going to be using in order to analyze our object. And so, right, you can obviously bring in your own file as well. And so we're going to be clicking on compare chemical shifts. So once you do that, you see we've populated the error table below, which is organized by a number of metrics. And so we can see here that if we click on one of the rows, that our chemical shift table is actually now also populated, and our primal object pops up on the side. So on the top of the error table, we see different labels. So state is going to be referring to the state of the structures. MA is going to be referring to the weighted mean absolute error. R is going to be related to the Pearson's correlation coefficient. RMSE weighted root mean squared error. And then BME is Bayesian maximum entropy weights. And so in PyShifts, assigned confirmation weights are attached to each primal object. And so we can see now that if we click on one of the rows of the chemical shift table, what is actually going to occur is that parts of the primal object are actually highlighted corresponding to the row. This makes it obviously easier to see on the object and easier to navigate. Now, the spheres on this object are going to be referring to the magnitude of difference, and so the size correlates to the magnitude of difference between the measured and comparison chemical shifts. And the color is going to be referring to the negative or positive difference, with red referring to the negative difference, and blue referring to the positive difference. Right, and so we can also see here that there are different ways to sort our error table, going over by the metrics we just went over. And so right now we sorted it by the mean absolute error. However, we can also sort it by the Pearson's coefficient correlation coefficient. And so right here we can see now that instead of being organized by the mean absolute error, we've organized it by the Pearson's correlation coefficient. And then we can also obviously do this with the RMSE, state, or weight as well. We can also look at different nucleus types. And so in this case, we're going to be looking at proton chemical shifts. We can also obviously look at carbon or nitrogen as well. And so once we run that, right, we can see that we have our proton chemical shifts. Right, and so finally now we're going to be going over sort of the advanced options within PyShifts. And so if we go here, you can see that there are a number of different ways we can actually use this platform. And so we can change the sphere size, the colors we'd like to use, the number of models we'd like to display, clusters, accuracy hits, all those things. So in this case, we'll also be going over the save function as well. So just as an example, I'm going to change the number of models I'd like to display within the platform. And then we're going to go back to error analysis. Right, and so we can see that once we actually run PyShifts again, so this may take a second. Right, so what we can see here now is, like so, our 20 different models have now popped up. And so that's one of the ways we can use the advanced option function of PyShifts. And so what we also have the option to do is save to file. So we can save to file our error table, our chemical shifts table, and then also this session in general. So if we'd like to save the 20 models we have available right here, and instead of having to go back and pull all this up, we can just save it to session. So if we click on this button, I'm going to save it as session save .pse. And so, once I do that, I'm now going to close out of PyShifts. Alright, so what I've gone ahead and done is I've closed out of PyShifts and I've actually pulled up PyMol again on the side here. So if I bring up my save session, session save PSE again, and I enter that, you can now see that the 20 models we've had pull up. And so these are just a couple features of PyShifts within the program, and obviously there are many more. Yeah.